In this video, I'm going to discuss about this polysaccharides from the chapter Biomolecules. Yes, first of all, the polysaccharides. The polysaccharides are nothing but the carbohydrates here, which are consisting of or which are made up of monosaccharide units. Yes. So on hydrolysis of these polysaccharides, in other words, will yield a number of monosaccharide units. That means in these polysaccharides, the monosaccharides units are connected by means of an oxide linkage, or also called as glycosidic linkage, by the removal of this water molecule. So a polysaccharide is nothing but a carbohydrate which is containing a large number of monosaccharide units connected by means of this glycosidic linkage or oxide linkage by the removal of the water molecule. Yes, we'll study about the starch, cellulose and glycogen in these polysaccharides. First of all, the starch. Yes, starch is the main storage unit in plants, and for human beings, where do you find this? Actually, is a source of it is cereals, roots, tubers, and some vegetables, and so the human beings find these polysaccharides or get this starch in the cereals, roots, tubers, and vegetables, and therefore it forms this major dietary source for human beings. So what it is made up of, what are the structural units of the starch is the glucose. What type of glucose? It's an alpha glucose. And if you see the structure of alpha glucose is nothing but a 6-membered ring. I'm writing the Harvard projection formula of it, where one of it is an oxygen, and draw these lines across this one. So at the, number these carbon atoms as 1, 2, 3, 4, 5, and 6 the carbon atom. So in alpha glucose, how this is find this alpha glucose, where H on the top, OH at the bottom of the Harvard projection at the carbon number 1 is nothing but the glucose. So rest all is the same. If OH is on the top, H is bottom, then it is called as beta glucose. So I'm writing the structure of alpha D plus glucose here, and OH, H, that carbon number 3 is a reverse of this configuration, that is H and OH, and rest all is the same, and the 6th carbon atom is CH2OH. So this starch, the structural unit is nothing but the alpha glucose units here. So there are 2 components we can find in the in this starch here. There's amylose and amylopectin. Here, let's look at the structure of this first amylose. Yes, amylose is nothing but, if these are the monosaccharide units, it's round ones, these monosaccharide units are linked together again by means of glycosidic linkages. So here it is a straight chain. So the monosaccharides are linked in the form of a straight chain here, unbranched chain. So it is not at all branched. There is no branches. We can see it's a straight chain, as we can see here. And this one, monosaccharides units are nothing but this alpha T plus glucose units here. And how these monosaccharide units are linked by means of C1 of one glucose unit is linked to the C4 of another glucose unit. So it is what type of linkage here? C1, C4 glycosidic linkages are formed here. And this forms only 15 to 20% of starch, and rest 80 to 85% of starch is nothing but amylopectin. So this amylose, as we can see, is a water-soluble component which forms 15 to 20% of the starch, and this is a straight chain, that is unbranched chain, and these monosaccharide units are linked by means of this C1, C4 glycosidic linkages. So I'll show you how these are linked here. Yes, if you can see, it is C1 and C4 glycosidic linkages. So this is one glucose. So first I'm writing the structure of glucose, that is alpha glucose. See, this is H, alpha is HOH here, and second carbon atom also the same thing. So first number them 1, 2, 3, 4, 5, and 6. Third position, it is reversed. Fourth is also the same thing. Yes. This is a structure of one glucose unit. I'm writing the structure of another glucose unit here. Again, the same thing. So HOH, alpha glucose again. So this is 1, 2, 3, 4, 5, and 6 number. So rest all I'll fill it up. HOH, OH, H, HOH. So one more glucose unit I write here, the same glucose, that is alpha with 1 oxygen. Number them 1, 2, 3, 4, 5, and 6. Filling up H and OH here. So what are the linkages we find here? C1 of one glucose is linked to the C4 of another glucose. So here C1 carbon atom of one glucose is linked to the C4 carbon atom by the removal of this water molecule. So this is how the water molecule is removed, and these two carbon atoms are linked by means of oxygen. Again C1, C4, this water molecule is also removed. So I'm removing that water molecule, I'm not writing it again. So how they are linked? By means of So this is the oxide linkage, as we can see here. Right, again oxide linkage. Right, so this goes on. So here again the C1 linked to C4 and this C4 linked to C1. So I'll show you that. So this oxygen is linked here. This goes on. Yes, this is how you can show the linkage. And what is the glycosidic linkage here? This oxide linkage is nothing but the glycosidic linkage here. So the C1 of one carbon atom is linked to the C4 of another carbon atom by means of this oxide linkage by the removal of this water molecule. This oxide linkage is called as glycosidic linkage, as we can see here. So this is what is amylose now? Amylose is a straight chain without the branches of alpha D glucose units, which are linked by means of C1, C4 glycosidic linkages. And let's see the structure of amylopectin now. This amylopectin here, that is amylose is water soluble. This is not soluble in water, which constitutes about 80 to 85% of starch. And again, this is also made up of alpha D glucose units. But here, if you can see here, it is these here. This is long branched chain. So the chain is, it's a branch chain. The straight chain again is made up of, these are the monosaccharide units, which is made up of C1, C4 glycosidic linkages. And whenever there is a branching here, these are the branches you can see here. So these, whenever this is the branching, therefore C1, C6 glycosidic linkage, which we can see here. Yes, whenever there is a branching, so we can see that C1 of this is linked to the C6 of this branch. Right. So let me write the structure of that. See, first of all, I write a straight chain and then do the branching for it. So same as what I wrote for amylose, same thing here. All right, the 6-membered ring with oxygen, and just number them 1, 2, 3, 4, 5, and 6. 1, 2, 3, 4, 5, and 6. 1, 2, 3, 4, 5, and 6. So again, this is what H here linked by means of oxygen. Here also linked by means of oxygen. And if I fill this up, this is H, OH, OH, H, H, CH2OH. Same thing here, H, OH, OH, H, H, CH2OH, H. Again, H, this is continued, this long chain, and H, OH, yes. So this is what we see in the straight chain. Whenever there is a branching, when is the branch? C6 of this is linked to the C1 of another one. So I'll write here with the C1 carbon atom here. So this is oxygen. So H, OH, H, OH, OH, H, H, OH, CH2OH, and H. So this is first carbon atom. So here, this OH of first carbon atom is linked to the, the carbon atom 1 is linked to the carbon atom 6 of next to glucose by the mole of, again, water molecule. So this is a glycosidic linkage we can see here. So I'm erasing this so that it can be shown easily. So here, yeah, this is how you show the linkage. Right, so properly. Yes. Right. So here, as we can see here, whenever there is a branching, so which carbon atom? First carbon atom and sixth carbon atom of the next glucose. And whenever there is a straight chain, first one and fourth one. So C1, C4 for straight chain, C6, C1, C6 for branching. Whenever there is a branch, we can see C1,6. Whenever there is a straight chain, we can see C1, C4. So this is the structure of amylopectin. So, and what is the difference here for amylose and amylopectin? I'll write here. So whenever this is amylose and this is amylopectin. So simple here, when this amylose, it is, as I said, water soluble, because we cannot see any branching here. This is simple structure, so this is water soluble. This is water insoluble because it is branching, we can see here. This forms about a very small percentage of starch, 15 to 20% of starch. This forms about 80 to 85% of starch here. Right. And as we said here, there is no branching we can see, only straight chains are formed. And here C1 of one glucose is linked to this carbon atom 4 of another glucose. So what we find here, C1, C4 glycosidic linkages in the straight chain. And here we can see the branching here. So whenever there is a branching, we can see, whenever there is a straight chain, we can say C1, C4 glycosidic linkage in straight chain. And whenever there is a branching, we can say C1, C6 glycosidic linkages. So in branches. So this is the difference in between amylose and amylopectin. So this is amylose, simply a straight chain where C1 of this thing is linked to C4 of this thing. And here it's a branched chain. So whenever there is a branching here like this, we can see that whenever there is a straight chain, C1, C4. Whenever there is a branch here, C1, C6 glycosidic linkages we can see here. So this is the difference in between the structure of amylose and amylopectin. Let's see the structure of cellulose. Yes. So what is the cellulose? This is found exclusively in plants. Only in plants we can find. And the cell wall of this is made up of what? The nothing but the cellulose here. And what is the structural unit of the cellulose? It is beta glucose units. And again, what is the linkage? Again, the oxide linkage or the glycosidic linkage, where C carbon number 1 of one glucose unit is linked to the carbon number 4 of another glucose unit. So what are the structural units of this? This is a beta glucose. So what is a beta glucose? I'll write the structure of beta glucose here. So beta glucose is nothing but alpha. This is again 6-membered ring with oxygen. Alpha, as I said, is H is on the top and OH at the bottom. So just the reverse of it. OH on the top, H at the bottom is nothing but the at carbon number 1. So is nothing but what this is beta glucose. And this is the structure of beta glucose. As we can see, it is C1, C4 glycosidic linkages. This is the carbon number 1. So this is what beta D plus glucose units. So 1 is linked to the 4 of another carbon atom. So this one. Again, oxygen. I'll write the same thing here. So this is carbon number 1, OH on the top, H bottom. Third carbon atom, fourth carbon atom, and fifth and sixth. So next glucose also I'll write here. Yes, your oxygen. Yes, this is the first carbon atom, OH, H, H, OH, OH, H, H, OH, CH2, OH, and H. Right. So if I show the glycosidic linkages here, see, here C1 of this and C4 of this carbon atom, again C1 and C4. So what you can see here, C1, C4, C1 of one glucose unit linked to the C4 of another glucose unit by the removal of water molecule, that is glycosidic linkages which are being formed here. So erase this, form the glycosidic linkage. So linked by means of this oxygen. So here, this oxygen, these are linked by means of this oxygen, and again here these two are linked by means of this oxygen. So this is how you see the glycosidic linkage. This is one glycosidic linkage, this one is another glycosidic linkage, and this goes on. So I can show it further with the oxygen itself. So erase this also. So this goes on, right? So oxygen. This is again oxygen. Right. So we can write the glycosidic linkages like this. And what are the structural units here? Beta D plus glucose units, which are linked to one another at, by means of glycosidic linkage in between C1 of one and C4 of another. So what type of linkages? C1, C4 glycosidic linkages we can find in the cellulose, which are exclusively found only in plants. So the cell wall of the plants is made up of this cellulose. Yes. And when we come to the glycogen here, this glycogen, yes, the carbohydrate is mainly found in the animal. So in the animal body, how these carbohydrates or polysaccharides stored in the form of glycogen. So whenever the body needs glycogen, it is broken down to simple glucose units. Whenever the body needs glucose, what happens? Whenever the body needs glucose, this glycogen is broken down. This glycogen is broken down with the help of the enzymes which are present in our body and will give this glucose which are necessary to the body. So it is called as animal starch because this is the resemblance or the structure of this glycogen is very similar to that of this amylopectin. So structure of this is very similar to that of this amylopectin. So we are calling this as starch. The starch, what means animal starch, the starch which is present in the animal. So animal starch. Right. So where do you present, where is this present in the animal body's livers, muscles, and brain? Yes, this is about this polysaccharides. If you like this video, please do like, share, and subscribe my channel. The whole chapter of this biomolecules has been discussed on my YouTube channel, and the details of this, and the link of this is given in the description box below.